Welcome back to the COVID-19 Diary 3rd edition. Um, this edition is about vaccine development, COVID-19 variants, and herd immunity. So the COVID-19 program is here to bring you information to empower science journalists on how to communicate matters on COVID-19 and also demystify our perceptions and make things clear out there. And this time it's on vaccine development, variants, and herd immunity. And I'm glad to be joined here by my wonderful, eloquent, exquisite co-panelists. Right close to me is Imelda Giselle Muji. Imelda, how are you feeling today?
I'm feeling very good.
Splendid. And also on the far side is Isabella Tata Henoko. Bella, how you doing?
I'm fine, thank you.
Great. So we just have a little time to just tell everyone joining us today the expectation. What is boiling in the COVID-19 Diary 10th edition and what's in for the people out there?
Alright, so with this third edition, we are going to talk about the COVID-19 variants, as you earlier said. You know, virus mutates as time goes on. So today we are going to know why they mutate and then the kind of variants that are out now. And then we're also going to talk about the vaccines. A lot of people do not know what the vaccine comprises of. They don't even know the name. All they know is they said they should go for vaccine. They are going for the vaccine. So today we are going to talk on that as well. And then we move on to herd immunity. A lot of people do not know what herd immunity is. So today we are going to learn what herd immunity is.
So that's the matter we have to really point out. Melda, what's your take on this one?
Okay, my take is for the herd immunity. I really want people to pay attention so that they will be encouraged to take the vaccine for our own safety. And also with the vaccine, it's good to know how vaccines are developed so that next time when we approach, we are not scared, nor even give out information that are irrelevant and not facts. So today, I'm sure our participants will learn a lot before they leave this session.
Great. So you had it. Summary is just for you to pay attention and also be part of the discussion all throughout. I wouldn't want to miss words. I'm going to invite the Director for ICT Knowledge Management Services here at the Association of African Universities, the person of Ms. Lidward Lamini, to give us her opening remarks.
Good afternoon from wherever you are connecting from. You are most welcome to the third episode of the COVID-19 Diary. We welcome each and every one of you. Thank you for your interest and your commitment to join us in this important episode where we will be discussing about vaccines, variants, and herd immunity. For those of you that have joined us in episode 1 and episode 2, you will agree with me that these sessions are extremely useful for us as we learn how to cope with living with the COVID-19 pandemic and its impacts. My name is Nodumo Dlamini. I'm the Director of ICT Services, Communications and Knowledge Management here at the Association. We are grateful to the support that we got from the National Research Foundation to launch this project. We have learned quite a lot through its implementation. We have also been able to engage a diverse number of participants and type of stakeholders in making sure we profile the issues around COVID-19. We also believe that we've also contributed to advocacy and dissemination of information to different types of stakeholders through these episodes. Today we've lined up for you a very important and very knowledgeable panelist speakers that will He is well published. He has mentored many students, both at PhD and master's level, and I am sure that you will enjoy the remarks from him. So Professor Olushola Oyewole, you are most welcome to address our distinguished participants that I see have connected from all the 5 regions of Africa and also from outside the continent. Thank you very much. I wish all of us a very successful event today to the 3rd episode of the COVID-19 Diary. Thank you.
I'm glad to welcome all of us to this special event focused on sharing our views on vaccines, variants, and herd immunity. We are glad to note that the world has risen to the challenges of the COVID-19 pandemic through the use of vaccines. One thing that the pandemic has taught us in Africa is the need for African countries to develop their capacity to produce vaccine. I'm glad to know that some African countries have risen up to this challenge, and they are working on developing their own internal technologies for vaccine production in their various countries. As we talk about the use of vaccines in Africa, people have been concerned about the effect of mutation on the vaccines that are being produced. The question that comes to mind is that people are asking, will the variants that are coming up as a result of mutation of the viruses be challenged by the vaccines that have been produced already? I'm glad to note that these are the issues we've brought to this platform to discuss today. We are glad to invite some specialists in Africa who will help us to discuss these particular issues. Let me welcome to this platform Dr. Elumono Richard Blyden, who is the founder and CEO of Every Biofarm, and also Professor Potiano Kalebu, who is a professor of immunovirology, who will be speaking from the Uganda Research Unit. Before I leave this opening speech, permit me to appreciate the COVID-19 Diary team at the Association of African Universities. I believe that with these additional platforms that have been provided, many Africans will know about vaccine production, the variants of the virus that are being produced.
Welcome back, our viewers. Before we continue, we'd like to talk about the previous episodes we've done on the COVID-19 diary. In the first episode, we actually tried to break down how COVID-19, actually the SARS-CoV-2 virus, it's been formed, and how viruses are formed and how they find themselves in us. With us, I would like to ask my colleagues if they can actually remember some of the things we spoke about on the first episode of the COVID-19 diary.
So you know, it was very exciting with the first edition where we understood the origin of SARS-CoV-2. We realized that the SARS-CoV-2 virus, the coronavirus, are quite a number of viruses together, lots of them. And some of them have emanated many years ago with strange names like the HUK1, NUL, lots of them. And those were the mild type, not so strong in terms of effect. But you know, somewhere in September 2011, 2012, 2009, there was the first one, which is the Mediterranean respiratory syndrome as a MERS. That also had some effect. Then we had the SARS. Then later on, SARS-CoV-2 here, the viral SARS-CoV-2. We appreciated how the SARS-CoV-2 has become a formidable virus compared to the previous viruses, the MERS and SARS. Because the SARS-CoV-2 has not been able to extend its mutation. Now it has variants, unlike those ones which cause just influenza and just cold. And that is where we began to appreciate how distinct the SARS-CoV-2 was and how scientists were looking for ways and means to understand how it was transmissible from animals to humans. Lots of perceptions were out there, but we did very well to provide solutions that it was a natural virus, not an engineered virus, which
many thought that it was from the factory or let's say the laboratory. It was a normal virus that existed with animals like camels and bats. And through other human activities and some activities beyond our control, these viruses gain transmissibility into human beings. And that's how we get the virus and how it affects us when it enters our human body. I want to throw that to Bella to explain how devastating the virus could be if it enters your body.
All right. So on the first episode, I remember I talked on that. I used the respiratory system to explain on how you get the symptoms if you're infected with the virus. I remember I said, you know, when we breathe in oxygen, it passes through the bronchi, then passes through the bronchioles, then to the alveoli. Now, when you're infected with the virus, it actually binds with the host cells in your body, and these cells are in your throat. They are in your throat. So when the virus enters, normally your immune cells is going to fight the virus. But then, if your immune cells is weak, how can you develop weak immune system? That is when you smoke alcohol, you take in more alcohol, or when you have underlying disease like diabetic or hypertension. Those causes your immune cells to be weak. So when that's happening, it's not going to fight the cell. Rather, your cell will be dead. And then instead of your alveoli to contain air for you to breathe out the carbon dioxide you are breathing in oxygen, it will rather be filled with the dead cells, and then it will block air from entering. That is when you feel that you can't breathe, you are short of breath and dry throat and all that. So this is how, when you are infected with a cell, it's going to you are going to develop those symptoms. And also we learned how when the virus enters into your body, it's going to combine with the host cell. There's an engine in your host cell called ribosome. So the virus enters in, it binds with the entrance of the host cell, which has a spike. I forgot the mention. So then it has a mechanical things it does, and then the RNA in the cells enters into your host cell, binds with the ribosome, and then replicates itself to develop more virus, and then destroys the cells, and then moves to another cell. So these are some of the things we learned on that.
You know, notwithstanding what Bella you said, you know, we also highlighted the enzyme ACE2 and the very role it plays. That, I mean, to my surprise, it shouldn't be so, but it's unfortunate that ACE2 is compromised at the receptor entry. To think that the viral genome of the coronavirus entering the cell is just a normal RNA visiting the DNA in the nucleus, and by so doing, the camouflage deceives the receptor. ACE2 is now fighting against its own self because it has been compromised, and that is one thing I was really sad. I was saddened though, but anyway, if you're infected, that is what happens to you.
And I actually like the fact that we actually differentiated with the RNA and the DNA, how they function separately, then hand in hand. For the DNA, I quietly remember that one actually transferred genetic information, and the RNA transformed that information and processed them. Okay, and going on to the second episode, we spoke about the myth versus the science of three main topics. That is the mystifying myth of microchip theory and antibiotic treatment. And with that, we did justice to it. I don't know if you remember anything or you want to say anything about that.
I remember Dr. Aaron Tetahinaka talked on that, and then he made us know the stages the virus, the vaccine goes through before it is before it is distributed.
Approved,
you know, there was a myth that there is a microchip in it, and then they are going to track us, our movements and all that. But he made us know that it passes through a rigorous process before it comes out. He talked about the R&D stage, which is a research and discovery stage. From there, it moves to the clinical, non-clinical studies, where the vaccine is tried on animals, like monkeys, to know the effective and the efficacy of the vaccine. And then from there, the scientists will submit their findings to the FDA in a form of IND, which is investigational new drug. From there, then they try it on humans, that is clinical development studies. They first try it on a few number of people.
Yes.
Then move to the larger, medium number of people, then to the larger number of people. From there, then they submit all their findings to the FDA again
for final approval.
Exactly.
Yes.
And this stage is the biological license application.
Absolutely.
Here they are trying to apply for their vaccines to be distributed to the public. Even that, FDA will send people. They will send people to the manufacturing place to. They'll search the place. They'll get to know that if this place is safe to develop a
All
vaccine.
the quality checks.
The chemistry they're going to use, the manufacturing process. I mean everything. They are going to check before they will give you the permit to distribute the vaccine. Now you are saying that they've put a microchip in it. First of all, the scientists who developed the vaccine, they don't even know where the vaccine is going. There's a pandemic. They are trying to block this pandemic. So they try and then develop a vaccine. They don't know where this vaccine is going. So how are they going to track you? And then in what stage are they going to put the microchip inside?
Yeah.
Do you get it?
He highlighted that, yes.
Dr. Mrna actually
Did justice to
yeah,
that.
he really did justice to that.
And at the end of the second episode, the misconception about the microchip has been ruled. And also we spoke about the demystifying myths, the lab and the development and accident theory. Ajima, do you remember something?
Well, I mean, it was sounded clear that, you know, the coronavirus compared on two facets. Either it was engineered in the laboratory or it was from its natural point through, let's say, zoonosis, through it being transferred from animal to human.
Humans.
And we weighed the options. We understood that the virus has features. Its genomic features are natural. And, you know, compared to an engineered virus in a laboratory would not look like how it looks with the kind of spikes it has. These are not engineered viruses. They wouldn't look like that. The World Health Organization have done the check, and the committee of virologists have done their checks and realized that the virus in its natural genomic state doesn't represent a virus that was engineered in a laboratory. I think that sounds it with the WHO. So we can move on the next one.
And the last demystifying point we actually spent time on was the mRNA and the DNA conspiracy theory.
Sure.
Yes.
Well, what the speaker, I remember it's a she gave a great
Jessica.
a great example using a padlock also.
Yes.
Like we are saying that when the virus enters into your body, it's going to change your DNA. She actually used a padlock with a different lock key. It's not possible to unlock that. So actually, if I'm going to base on that example, she's trying to say that there's no way the virus is going to change your DNA.
And with our first episode, we clearly stated that RNAs and DNAs are two different entities. They work hand in hand, but they cannot work for each other, but they help each other to achieve some result. Now, in our third edition, we are going to spend time talking about vaccines, variants, and herd immunity. With these three topics, we have special people who are well vested to help us understand these three things. And we are going to go home with a lot of knowledge. And then I know there are scientific facts and all the misunderstanding and the information we get out there, we are going to feed people now with the right information. Please stay with us.
Welcome back, and I would like to put in an
Apology for the sound glitches that you had earlier with the recap of the previous episodes. I hope our apology is accepted. We'll have to now move to the main business of today's COVID-19 Diary third edition on COVID-19 vaccine development, variant, and herd immunity. And the first person to be introduced to speak on COVID-19 vaccines development is Dr. Mrna Richard Blyden, and he is the founder and CEO of Avril Biopharma and Incorporation and the mother of Best Initiative. He's going to help us understand the composition of vaccines and how they are developed, the stages they go through, and why you should trust the vaccines after the rigorous procedures they've been through. So if you can hear me, Dr. Blyden, I would like to invite you to take your post. It's your time. Let's hear from you, Doc. Yes, loud and clear, Doc. Absolutely.
definition of who was this character called Sakpata, different names, Babalu Aye, and so forth. They're all the same character in different parts of Africa, from as far as Guinea, Freetown, all the way down to Angola. Various forms of this character exist. And like all African deities, he had a character, and his character was that he could harm you, but he could also heal you. He could harm you, but he could also heal you. If you think about that, that is the foundation principle of immunology. And what we discover when we go back to the original sources of vaccination, inoculation, variolation, you'll hear all these terms. But when you go back to the original historical sources about the discovery of smallpox immunization, what you find is that, in fact, the information about immunizing against smallpox emerged from African traditional medicine. Here's one example, a very interesting one. Again, adds another dimension to our thinking about why we need to develop our capacity for biomanufacturing in Africa now. This dates back to the year 1716. And it is a quote from the Royal Philosophical Transactions of London. So I guess at that time, that would have been the premier, the highest level of scientific source that was available. And what it says is that, in fact, Africans on the west coast of Africa, where at that time, of course, slavery was at its height, on the coast of Guinea, as it says, if the traders, the slave ship captains, happened to take on any captives that had not had smallpox, they sent them back to the shore to be inoculated so that they would make a better market in the West Indies. In other words, the practice of immunization was widespread enough in West Africa as a public health preventative for it to be of economic value to the slave traders. It earned them a premium when they got to the New World because they could guarantee that their cargo was not going to be sick and have smallpox. And there are many implications of this we don't have to go in time to go into today. But I did want to put up some of these very interesting primary sources of information for you to research on your own and get a sense of it. So the thing about today is that it is an opportunity for us to try to identify what are the best technologies for Africa today that could create viable strategies for not only potentially fighting this current pandemic, but also preparing ourselves for future pandemics so that we don't find ourselves in a situation where we're importing 90% of our pharmaceutical products and they're not available during a pandemic because of disruptions. And the theme of that is that indeed healthcare is wealth care. If you're able to maintain a healthy population, then that population is going to be able to afford better healthcare. It's what you call a positive feedback loop. And that is what we have to try to design for the future when we talk about designing vaccines or developing vaccines. So I start with this slide to really give you a sense of what we've learned about that challenge for the future. Of course, we all know now about variants and the fact that the virus behaves differently in different countries. It arrives in different countries at different times. And we also know that we have a distribution technology to really be able to think of a situation which we can respond to pandemics essentially on demand, in the same way that we can go online and we can pick and choose color, shape, and size for our Nike shoes or whatever other product we're buying. And then we click the button and the product gets delivered to us by mail. Well, we would love to be able to do that with vaccine. Would be lovely to have our Minister of Health be able to log in online, say, okay, these are the variants we have in our country today. This is the kind of production capacity we have. These are the strains that have been circulating, et cetera, and then be able to actually order a vaccine that is tailor-made for his own local population. Well, for years, I've had this set of slides and I've shown to people and they've nodded their heads. But, you know, this pandemic, we saw something really spectacular, which is we saw this principle in action. The government of South Africa was able to make a strategic decision that they were not going to use the AstraZeneca vaccine, which they had already bought, because their scientists had been surveying the populations, and they decided that that vaccine in South Africa was not going to have a high enough level of efficacy to make it worthwhile for them to spend that money and on distributing it. So we come to this point about how important information is to all of this. If we're going to develop a vaccine or any kind of drug, we need to know about the pathogen, the agent that's causing it. In this case, let's say SARS-CoV-2. And we need to try to identify not only where it is today, but also maybe where it's going to be in a week from now, a month from now, a year from now. And we've all had this experience in real time over the last year or so. But I show you this picture here to show that when we are considering developing a product, we have to consider epidemiology, evolution, all of these factors. And in September of 2020 was when the first variant emerged, and that was the Delta variant in South Africa. All of the dots that you can see on these graphs are basically different variants that were there at the time. There are many more now, of course. Each one is really a variant. However, the only one at that time that was emerging as a variant of concern was the South Africa variant, the Delta variant. There are lots of other interesting things about these graphs I'm not going to go into here, but suffice to say that by getting information from the genetics of the virus, from the DNA sequence or the RNA sequence of the pathogen, the virus pathogen, we are able to understand not only how to tailor make the vaccine to it, but also to see, how to match it up with real world concerns such as climate, geography, population, all that sort of thing. So let's take a little bit of a deeper dive here. This is the analysis that we did with our own technology, which I call structural genetics, something that I created. And it's a method of analysis that gives you a unique and different perspective on the evolution of the virus. In the left-hand side panel, all of the little black dots represent all of the variety of coronaviruses. We've heard again a few of those about those today. The two clumps at the bottom there, one of those is MERS, the other one is SARS, and all of the brown dots are SARS-CoV-2. And you can see that it emerged very rapidly and diversified very quickly. And even when you look at the zoom close up here, that's just focusing in on the situation in South America, Brazil, Venezuela, Mexico. You can see that even after only about 9 months or so, the virus was quite diversified into these two clusters. So the basic principle then, to summarize, is that we want to be able to go from some sort of informatics analysis with the computer to actually developing a live product that's responsive and that we can give to the population to protect them. And over the years, I've been developing a technology that's based on the simplest possible platform, because in Africa, I think our objective is to say, well, what is the platform that we can use? We don't have pharmaceutical industries, which is why we're importing all of our medicine so far. But what industries do we have? Of course, we have agriculture in Africa, we have mining, we have Information technology is very well developed, as we see from the South Africa case. So how can we find technologies that we can use to make vaccines, but that can be based on our existing platform of the economy that we do have? So what I have done, again, the technology that, the enabling technology that I've been working with and working on for a number of years is to use eggs as a miniature bioreactor for making all sorts of biopharmaceutical products, but in particular, obviously, vaccines. Eggs have been used for a long time. In fact, eggs have been used to make vaccines in Africa since 1932 at places like the Institut Pasteur in Dakar and in Tunisia and one or two other places in Africa. They're used also to make veterinary vaccines till this day. And so it's a very, while the rest of the world looks at eggs as a somewhat old-fashioned technology, that might be your reflexes to say, well, this is old-fashioned, or we want mRNA, we want the latest technology. In fact, I'm going to persuade you today that this does represent the latest technology. And the reason it represents the latest technology, because this is true biotechnology. The key things that we really want to happen in Africa, however, is that we want to be able to support the manufacturing of our products with a local supply chain of materials. We've learned from this pandemic also that it doesn't matter how efficient how beautiful big factory you have, if you can't get the raw materials, you're not going to be able to make the product. This happened in several places. In Brazil, for example, they had signed contracts with Sinovac and everything to make, they'd started to build a new factory to make a COVID-19 vaccine, emergency. But when they looked at the details, they realized they would not be able to get the raw materials they needed to make those vaccines. And so they had to cancel the project. So how do we overcome that? Well, we use the egg because everyone can raise chickens, and those chickens can lay eggs and produce eggs. So in the very worst case, we know that our raw material will always be available. The role then of the government or the sponsor is to provide a platform of egg production so that we can use these more advanced technologies involving the recombinant protein expression technology to use those eggs to make vaccines. It turns out that with the technology I've developed, you can make not only the simple subunit vaccines by just making the spike protein or some part of the virus. That's a common way to make vaccines. But there's a variety of other vaccines that can be made, even including mRNA vaccines based on some of our early data that we have now. So there are great prospects. And what we would like to do is really put this into a context where the academia of Africa can innovate different types of vaccines, and we can provide a platform across the entire continent where those products could be manufactured using local materials. And all that the factory needs to receive is an email with a DNA sequence in it, and they would be able to produce a vaccine or other product, or an insulin, or a diagnostic test, malaria vaccine. The future is endless with this type of new technology. Just to demonstrate, this is our latest results producing the spike protein from SARS-CoV-2. Actually, two pieces of it, one the whole thing, and the other one is the small part of it that is binding specifically to the ACE2 receptor, which again you've heard discussed a little bit earlier.
Hello, Doc.
The way this is done is to make a vector that essentially takes a blueprint for this molecule. That's the cartoon illustration on the right.
Hello, Doc.
And to introduce that into the...
I'm afraid you have 2 minutes to wrap up, please.
Thank you. Introduce it into the egg, and the result that you get is the production of a protein that can be used to immunize people. To give you some idea of how effective that can be, we can get a lot of doses of vaccine from a relatively small number of eggs. I'm not suggesting that you're going to make the vaccine in the village, but this is the number of eggs you would need to produce to be able to make 5,000 doses of vaccine, just to give you some idea. Of course, as I said, there are lots of other products you can make. And if we invest in eggs or technologies that are based on local agricultural production, then we have an opportunity to also support the surveillance that it takes. We can make the same products I use to put in the test kits to test people so that we know what the status of the population is and so forth. And so that's why we've created the Mother of Birds initiative. And the idea behind this is to bring together all of the resources we have in Africa, from academics to industry to government, to actually set ourselves a challenge to innovate, clinically test, and manufacture a COVID-19 vaccine by the end of 2022, and then use that to build a network of capacity across the continent that can be used for the next strain or the next variant of the vaccine that comes along. In order for that to be successful, as I said, we need to come up with a concept of partnership, exchange of information, exchange of ideas, but ultimately that feeds into a production model that employs African farmers, that employs African doctors to do the clinical testing and so forth, and that relies on intellectual property that's created locally as well. So this is what I call the bottom-up strategy compared to the top-down strategy that is commonly been used in the past. So the top-down tech transfer model.
Thank you very much, Dr. Blyden.
Yep, I'll just round up now.
Yeah.
We've seen during this pandemic that the idea there is to manufacture a bulk product, let's say, by a pharmaceutical company in India or Europe or somewhere, and send that bulk product to Africa to be placed in the bottles of what's called fill and finish. So just the end part, the top of the pyramid, is all that has to happen in that. The problem with that is that, okay, you're sending a bulk product, but still you're still importing the vast majority of what you need. And if anything goes wrong with the pandemic disruption or war breaks out or something like that, then we're still back in the same position that we were in before. So to build a truly resilient system, we're going to have to build hen houses and raise our flocks to produce the eggs, process those eggs to make vaccines locally to support our public health efforts to fight pandemics and so forth. And these are the kinds of innovations that we hope you will join us in doing. And I hope out of this meeting, a conversation can begin towards that end. Thank you very much. I'll stop there. I think that's pretty much the end of the presentation anyhow.
Richard Blyden, for your presentation on COVID-19 vaccines development and also the novel means that you believe will be sensitive to the African continent. We believe that participants will be able to contribute and then ask more questions to its viability and sustainability as well. But we will have to move to our next presenter, who is Professor Pontiano Kalibu. He's a professor of immunovirology and also director of MRC, UVRI, and LSHTM Uganda Research Unit. Professor Pontiano, if you can hear me, it is your time to present. Absolutely, please. Go ahead. Yes, please.
Variants of the COVID-19 virus, SARS-CoV-2. My presentation, I assumed there's some knowledge about the virus, there's some knowledge about the variant. It's really refreshing ourselves. I'm not talking to a totally new audience about this subject. But all the same, I said, let me give you a brief just to understand a few concepts, what the virus is, the structure of SARS-CoV-2, the genome, how the virus replicates and mutates. I listened to the earlier conversations. I think you have had some of these conversations before, the lineages, the variants, a bit of our work we're doing in Uganda, and then this new Omicron and the future. I think the virus, this is not news to many of you, is an amazing tiny subject. People are saying, is it living or is it non-living? But the fact is that the virus is very tiny, infectious particle that can only reproduce by infecting our host cells. The virus will commandeer the host cell and use its resources to make more viruses, basically reprogramming it to become a virus factory. It's amazing how the virus replicates using our own cells for many of the ingredients it needs. It cannot live by itself like bacteria, and the host cell this virus can infect many host cells, animals, plants, etc. And there are mostly two types of viruses, one that has a genetic composition of RNA and another one which has a genetic composition of DNA. And I think we had some, you are aware of some of these. Now, before I really go deeper, this is something we need to remind ourselves about the virus. The upper one is the structure, and the structure gives us how the virus is made up, the spike, which is very important, which the virus uses to enter our cells, the nucleocapsid that surrounds our RNA, and in this case, RNA, single-stranded RNA for but also you need to understand the genomics, the genome of the virus. If you look at the RNA, it is a large genome, but we're interested in a few areas. When we do the sequencing, whether it's sometimes full genome sequencing or parts of the virus, we're interested in a few areas. But also we have to look at the whole virus, to look at all the mutations, the open reading frame. Plus a lot about the behavior of this virus in terms of transmission, in terms of many other factors. I don't want you to go deep into this, but just to understand the two different mechanisms a virus uses to replicate. If it's a DNA virus, it will enter into our nucleus, yeah, and then there will be transcription to a messenger RNA, and then, of course, the ribosomal RNA to start making the messenger RNA has to go outside. I don't know whether I don't have, can you see my pointer? Is it possible to see a pointer?
Yes, it is.
You can. The nucleus, and then you have the ribosomes, which some of you have been talking about, and you start making your proteins. The virus that has RNA does not need to go into the nucleus. It is in the cytosol, but it has to make, again, the proteins. Amino acids. And in that process of movement, if it's a DNA virus to transcription, RNA, or it's an RNA virus with all ribosomal RNA making the amino acid, there are mistakes that are made. And we generate mutations. And RNA viruses are very good at generating these mutations, and hence arising the many mutations, viruses, and variants that we have. Some of these can come due to pressure afterwards, immune pressure and the rest, and the virus finds a way of surviving. So the virus is very important. The idea for this slide is not go into the details, but to make us understand how we use our host cells and how the DNA and RNA virus differ in their replication. So these mutations do occur. They can result due to changes in the genetic composition out of a single base unit of RNA, but there could be also be deletions and insertions. And we see this in our variants. Some have deletions others have mutation insertions. And some of these bases lead to protein changes and hence the properties of the virus. When SARS-CoV-2 was first described very early on in China, two lineages were identified from the very beginning, lineage A and lineage B. And then later, through Rambert and others, they came up with a dynamic nomenclature that proposed some initial how to name these viruses, the A1, A2, B1. We still, at that time, had those variations of the A's and the B's, and these spread everywhere until December 2020, when we had the first variant of concern, which had so many mutations. It was initially called a UK variant, more than 17 mutations. And that was this.
Recording in progress.
Data, we have about
Recording
9 million,
stopped.
9 million genomes that have
Recording
been
in progress.
that are very low from Africa, about 1% from Africa. And the majority of these are from South Africa. So we need to do a little bit more. But as we when you look at new sequences now, Omicron is the dominant one. Omicron is now dominating all the new sequences. What are the variants of concern? These are the variants that are associated with one or more of the following changes at a degree of global public health importance: increased transmissibility, increased virulence or change in clinical disease presentation, decrease in effectiveness of public health and social measures, including vaccines that we're talking about. And hence, this slide summarizes the different variants that have been described. Some of them,
Recording
like
stopped.
the Alpha, which is called initially called the UK variant, is now de-escalated. And the reason it is de-escalated is because it is no longer circulating in big numbers. And I'll talk more about some of these that are being de-escalated. But as you can see, for most of these variants of concern, there's increased transmissibility, increased severity of disease, and very important, decreased effectiveness of the vaccines and neutralizing antibodies. And that has been increase in transmissibility. For instance, Alpha, when it was first described, it was about 50% more transmissible than the original Wuhan. Delta, more than 60% transmissible than the Alpha. And there was a higher viral load and longer period of PCR positivity. Omicron, more than 100% more transmissible than even Delta. Very, very highly transmissible. What is the impact of these variants? And I borrowed this slide from Slim and Tulio to show how with the introduction of new variants, the waves go up in India, in Brazil, Gamma variants, the infections go up. In South Africa, Beta and Delta, you see how it infects already the epidemic and the pandemic with the increase in number of transmissions.
Recording in progress.
Variants of interest. For these, compared to the reference isolate, their genomes have mutations with established or suspected phenotypic implications. Either they have been identified to cause community transmission or multiple CD4 COVID-19 cases or clusters. Otherwise, these have been described in consultation with the WHO SARS-CoV-2 virus evolution, looking at their mutations and all that. But we have seen now, as we move along, that many of these have been de-escalated because they are reduced saturation or no longer detected. And this slide shows you some of those variants of concern that we described earlier on, but are becoming less of a problem. In fact, the last one, the A23.1, was our group here was one of those that described it. It had the mutations. Some of the mutations were of concern, of course, had been described in some of the other variants of interest, and it was identified as a variant of concern. But even here, it's no longer circulating in Uganda. There are also variants under monitoring. And again, these are when sequences come, there's evidence, some can be strong or weak evidence to show that these may have properties that are similar to the variants of concern that have been described. And one of them is the one that was described in Congo not long ago, the B1.640 in September 2021. What have we done here in Uganda? We have been quite active together with our colleagues to look at the variants that we have. Initially, like many other African countries, most of the variants, the lineages we saw were lineages similar to where individuals came from. If somebody, as I mentioned, came from Europe, the lineages B.1, or B.4 were the most common. And then, as I mentioned, we described the variants A23.1, which became dominant later on. Recently, we have been looking at our variants and lineages. What you can see in yellow is the introduction of Omicron. Initially, it was among travelers that were coming in, but now it is widespread in our communities as well. We have used both genomic sequencing and PCR-based genotyping, which can be a screening. It's not really very accurate, but it can be a way of screening. Omicron taking off very, very quickly. That was from the beginning, from the end of 2021, November, December, added 2022, Omicron was dominant. If you look at our waves, the initial wave was mostly the A23, which I talked about. The second wave we had here in Uganda was mostly Delta. And the third wave we had, the last one in December and 2021 and early January 2022, it was mostly Omicron. So really, from 2021 to 2020, appears to be
Recording
even more
in
transmissible.
progress.
If you look at this slide, it's a bit now old, more than a month old, you can see globally how these sublineages are spreading. We still have the dominance of BA.1, but others are coming on. Others are coming on. Even within Africa, the BA.1, BA.1.1, and the BA.2. So Omicron has really taken on. This slide summarizes, if you compare Omicron with Delta, transmissibility, 3 to 4 times more than Delta. Disease severity, slightly less, which I think was quite good in that although more numbers were affected, but less hospitalization. And it's now even thought that 60% less risk of death. Some differences in clinical presentation, less loss of smell, increased sore throat. But most important, when you look at the receptors this virus is used, the S2 and of course the TMPRSS2 that is common in the lower tract, the Omicron appears to have a higher affinity for S2 than Delta, but much less affinity to this other TMP, which is more common in the lower tract. And that is the reason we see more virus maybe in the upper tract of the respiratory tract, and hence more transmissions because the virus can easily be transmitted, but less disease because there is less virus in the lower part of the tract. As far as vaccines are concerned, less neutralizable than even Delta. Most of these vaccines, the neutralization is not as efficient with the Omicron. You need a booster dose. But the good thing is that the vaccines can still prevent severe disease. Even individuals who have been infected with Delta before, their serum is less neutralizing the Omicron. The monoclonal antibodies that are being used for treatment, again, Omicron, unfortunately, is less effective, is less neutralized by these monoclonal antibodies. However, for the antivirals, the good thing, like remdesivir, at least it is sensitive. Diagnosis. We are able to diagnose most of the Omicron, but of course there are differences, especially if you have probes and primers that are in the spike. That's why initially in South Africa, they reported to us using the Thermo Fisher, the S drop, in that if you use some of these primers and probes, you may miss the Omicron because of the many mutations. It is possible if another variant will come. As we said, the virus does mutate. Of course, sometimes the mutations are good for where the mutation, where the virus will go. When Delta came, many virologists thought this is the end. If the virus mutates more, it will just burn itself out. Then Omicron came, but they had good things that Omicron came. It is transmitting very fast, but at least a milder disease than Omicron. Will the next variant be more mild than Omicron? You can't tell. Again, mutations can move in all directions. All we need to do is to be ready with their surveillance, but also to have the technology that has been talked about on vaccines to be able to address all these new variants. Will vaccines be effective for future variants? Nobody can tell. We have seen it that some of these vaccines are less effective against the variant. I think the key thing is to have the technology to be able to follow the variants as they come on. So I would like to end by acknowledging the colleagues who have been working with, but also those who have supported us in different ways. Thank you.
Professor Pontiano Kalibu, for your presentation, as well as Dr. Illuminano Richard Blyden for yours as well. And you know, the pickup is that even though we live in oblivion about what's next for the variant and the vaccines that could eradicate this variant, we look forward that we will be safe. So team, that is the interesting part of the whole episode, where we engage our participants to also share their comments and questions and put them to the various speakers. So if you want to put your question to Dr. Blyden, mention his name, that is to Dr. Blyden. If you want to put it to Professor Pontiano, just put it to him, and then we'll be here just to carry out the message on your behalf. So it's time for you to bring your questions. If you have a question, just put your raise your hand, and then the head admin is going to give you access to share your question and have it answered. Right. Whilst we wait for participants to get their questions together and then brought on board, let's take a short video of a survey that was conducted by the COVID-19 Diary team on the state of awareness on variant vaccine development and herd immunity right now.
I got the first shot and I am waiting for the second. I'm not sure what the name of the vaccine is. All I know is it's a COVID-19 vaccine that is given twice. The stories surrounding the vaccines are confusing, and I did not want to be confused. So I simply went with the one that was available at the time. I'm not sure what herd immunity is because it hasn't been made known yet. We, the people on the streets, do not hear about such things until someone hears about them and comes to inform us.
There's Johnson & Johnson, there's Pfizer, there's Mrna, but I've been vaccinated with Johnson & Johnson. So in a population, we expect a particular number of people to be vaccinated. A certain percentage of the population to be vaccinated. So I think when that percentage is achieved, then we are talking about herd immunity.
Have you been vaccinated?
No.
No.
Yeah.
Why?
I'm not ready yet.
So have you heard the name herd immunity?
No.
COVID no.
I have been vaccinated, but I don't recall the name or type of vaccines I received. After the jab, I had a bad experience. I couldn't move as I used to for 3 days, and I had hand discomfort and malaria symptoms. I was in so much pain that I decided not to take the second jab. But after hearing from others about how vital it is, I encouraged myself to do so and took the second jab. In terms of the variants, I've heard of them, but I don't know much about them.
I got vaccinated with the AstraZeneca. I know the Johnson & Johnson, the Mrna, and this other one, Delta Cron, and just playing with the words there. It is not the virus itself, okay. To go into scientific terms, we have something we call messenger RNA. So that is what they used for the vaccine.
Have you been vaccinated?
No.
Why?
Okay, I don't have any reason, but I just don't want to. Because I've been hearing rumors, some people will be vaccinated, but nothing will happen to them. Others to go for the injection and be complaining, my leg, this. So I don't know what will happen to me. That's why I've concluded not to vaccinate at all.
Have you heard of the word herd immunity?
Herd immunity? No.
I have been vaccinated twice, but the vaccine's name escapes me. I had pains all over my body for 3 days following the first shots, but I was okay after that. I wasn't alarmed when I started experiencing the side effects of the vaccines because I was already informed. I have heard of the variants, but I don't know much about them.
I took the vaccine, but I have no idea which one it was or what it is comprised of. After getting the COVID-19 vaccine, I felt ill, couldn't sleep at night, and experienced cold, but now I'm okay. After receiving the vaccine, I was terrified since I heard from people that I would die in 5 years.
I was given the Johnson & Johnson vaccine, but I have no idea what it contains. After receiving the vaccine, I developed headache and felt feverish. I had 2 doses of the vaccine. I remember my husband experienced serious symptoms after taking the vaccine, which include flames, headache, and fever. I have never heard of herd immunity.
I haven't been vaccinated because I'm afraid. I heard someone was severely sick and others were even hospitalized, which is quite disturbing to me. Therefore, I don't believe I'll get the vaccine.
Have you been vaccinated?
No, please, I've not. Due to some misconception about what people that are vaccinated, they're giving out, the kind of sicknesses they are having after being vaccinated, we are scared to be vaccinated too.
Have you heard the word herd immunity?
Yeah, I've heard it before, but actually I don't know what it is yet.
I received the first vaccine, but I don't remember what it is called. I felt sick the first day I was vaccinated and feverish as well. So I took paracetamol as directed, and the next day I was fine. I also heard from certain pastors that the vaccine contains 666, but I didn't pay much heed to it, just like many were skeptical when the late President John Atta Mills announced the yellow fever vaccine. But for me, it was quite beneficial. I was allowed to travel to another country with a card proving that I had been vaccinated.
So welcome back after the view on the survey reports that was conducted by the COVID-19 diary team towards the third edition. And I mean, you felt the bizarre comment that were made by some of the respondents concerning their ignorance to herd immunity and the vaccines and a whole lot. And you know, it's time that we introduce our special guest here in the studio to talk about herd immunity, a very important term to the COVID-19 pandemic that we have not really gotten so much information on. So I want to introduce Isabella Henokwa to take us through herd immunity right from the studios right here. Bill,
Hello.
It's your time.
Thank you very much, Mr. Ajumanchedakun. You're all once again welcome to the third edition of the COVID-19 Diary. My name is Isabella Tete Ahinakwa, and I'm here to present to you my knowledge on herd immunity. And I would like to share my presentation with you so that as I present, you go with me. Producer, please, can you share my PowerPoint? Okay. Thank you. Okay, so talking on herd immunity, with this presentation, I'm going to look at, I'm going to talk on immunity and the types. I'm also going to talk on how we obtain immunity. I'm also going to talk on how we calculate herd immunity threshold and also the challenges in achieving herd immunity. Now, to understand what herd immunity is, we first need to understand what immunity is. Now, immunity refers to the body's ability to repel pathogens such as bacteria, viruses, and germs. And then there are two ways to obtain immunity. The first one is obtaining it by birth. That is when a newborn baby obtains immunity through his or her mother, through their placenta. And that is known as the passive immunity. Now, aside that, we can also obtain immunity when the body produces new antibodies throughout our lifetime, and that is what we know as active immunity. Now, that can also be obtained in two ways. We have the natural active immunity and then the artificial immunity, that is also known as vaccine-induced immunity. Now, I'm going to talk on these two. When we talk of natural active immunity, this is when your body produces antibodies or becomes immune to a virus after you are being infected with it the second time. Now, when the body, how does this happen? When the body is exposed to the pathogen, a kind of blood cell produces antibodies that will fight this virus. Now, as your body is producing this antibodies, it also produces memory cells, and these memory cells can be in your body for a very long time depending on the type of the pathogen. And then when your body is exposed to the virus the second time, your immune system response is faster as compared to the first time you were infected with it. For example, as I said, this depends on the type of pathogen. When we talk, for example, measles or chickenpox, when you are infected with it the first time, there's no way you are going to be infected with, you are going to experience the symptoms when you're infected with it the second time, because naturally you've obtained immunity. Now, the second one is vaccine-induced immunity. That is when you gain immunity after receiving a complete dose or an approved or authorized vaccine. Now, how is this? Most vaccines use a dead form of pathogen to stimulate an immune response. So when your immune system detects this dead pathogen, it begins to take steps to destroy the virus in the form of antibodies and memory cells. So as I said, the previous one, the natural immunity, when your body is exposed to the virus, you gain immunity naturally. And I told you that as your body is producing antibodies, it also produces memory cells, and these cells are the ones that affect or that fights the virus the second time. That is how the vaccine immunity also works. But this time, it is not naturally. You've been injected with a substance, and that is what is fighting the virus when you are exposed to it the second time. Now, immunity does not only protect you from being sick if you are exposed to the virus the second time, but it also protects you from spreading the virus to other person. And this is a method of preventing you from spreading the virus to many people. When this occurs, that is when we are talking about herd immunity. So I'll go on to define what herd immunity is. Now, there are lots and different explanations for this term, But to simply put it, it refers to how many individuals in a country must be immune in order for the incidence of new infections to decrease. So we are saying that in a population, how many people need to obtain immunity for them to decrease the spread of the virus. Now, the proportion of a person must be immune in order to develop immunity, herd immunity varies depending on the disease. For example, according to World Health Organization, measles, we need to obtain about 95% need to obtain immunity before we will reach herd immunity. Also, in the case of polio, about 80% needs to be immune before we achieve immunity. But with COVID-19, we do not know the proportion that needs to be immune before we reach herd immunity. Now, how is herd immunity threshold calculated? We are saying that in a vulnerable population, some diseases are highly infected. For example, measles. Let's say if I am infected with measles, I can spread the virus to about 16 to 18 additional people. And in the case of flu, if I am infected with it, I can transmit the virus to 2 to 3 more people. So to calculate herd immunity, we are saying that the formula is 1 minus 1 divided by R0, where R0 refers to the average number of secondary infections caused by a single infected individual introduced to a susceptible population. So let's say if the R0 of a disease is 4, that means we are going to calculate it as 1 minus 1 divided by 4. And then the answer is going to be 0.75. That means we are saying 3 people out of the 4 needs to be immune, whether naturally or whether through a vaccination, before we can reach herd immunity. And also, if the R0 is 3, that means if I am infected with the virus and then I pass it to 3 people, that means out of the 3, 2 needs to be immune before we can say that we are achieving herd immunity. Now, to reach herd immunity, there are some factors that need to be considered, because if these are not considered, then we are not achieving the herd immunity. One is to check whether the vaccine prevents us from transferring the virus to others. If the vaccine cannot stop us from transferring the virus to others, then there's no way we are going to achieve herd immunity through vaccination by a proportion. We are saying that with herd immunity, a proportion of population needs to be vaccinated. But if the efficacy of the virus cannot stop us from transferring the virus to others, then the whole population needs to be vaccinated instead of some proportion of the population. Also, another challenge is the vaccine rollout. Is when the vaccine rollout is uneven. Some countries started vaccinating their population in late December 2022, and others started in mid-2021. Also, recently, according to a statement by the Multilateral Leaders Task Force from their 8th meeting on COVID-19, 23 people are yet to fully vaccinate 10% of their population, and then 73 are also yet to achieve 40%. That means most of us are still not been vaccinated. If this happens, we will not be able to reach herd immunity. Also, the emergence of new variants, as Dr. Kalibo presented, as time goes on, the virus is going to mutate, and we know about the Delta variant. We know about the Omicron variant. Some of these variants can dodge the efficacy of the vaccine. We are saying it can prevent you from getting sick the second time. But some of these variants are very strong to the extent that it prevents you from even getting, from even fighting the virus. And when this happens, the efficacy of the vaccine is not going to work. you obviously be infected the second time. And when this happens, we are not reaching herd immunity. And also, immunity might not last forever. Given what is known about SARS-CoV-2, whether natural or artificial immunity, it wanes over time. We are saying that you can naturally obtain immunity when you are infected with the virus. But then, according to research, we don't know how long this can last. You get it? So if you are naturally immune and then you expose yourself to be infected the second time, we don't know whether it's the memory cell is still in your body to fight the virus the second time. So in conclusion, I will advise that we all get vaccinated, although we don't know whether these the various vaccines produced for the COVID-19 can stop us from transferring the virus to others. But then we all know that it can protect us from getting sick when exposed to the virus the second time. Thank you.
Thank you very much, Isabella Tainoko, for your presentation. And you know, it's quite sad because we've seen unsatisfactory results with the African continent and the rate of vaccination. And I asked myself, how close are we to herd immunity? Your guess is as good as mine. Just give me a short while. Let me just mention out the questions that I've received so far. And then I believe that my speakers are attentively looking out for which one was addressed to them, and then they can answer within the shortest possible time so we can have a good wrap to today's meeting. The first one is from David Munyeri, and he says that why are African countries lagging behind in the production of vaccines? If at all its capacity, what does it take to train researchers? And if it's eggs production, why not encourage farmers to produce more eggs and have more vaccines? Second question from Dr. Anthony Donubari Anwen. Says, great presentation, very articulate organization of tests. Education and environmental quality have serious effect on healthcare delivery. Can local conditions affect disease variants, especially considering hygiene and climatic variables? That is from Prince James, and he says, the reasons for low vaccine production are varied and complex. First, technology required is complicated and expensive. For instance, most vaccines require freezing temperatures that require constant electricity supply. It is commonplace that electricity production on the continent is still low. And secondly, the geopolitical and geostrategic interests surrounding vaccine production. The third is patent rights, where it is not easy, for example, country A or X in Africa to produce a particular vaccine that is already patented by country Y somewhere else. And all the above make the global vaccines policies substantially rigid, making African countries' vaccines production next to impossible. However, Africa should raise a concerted voice in the worldwide bargaining and negotiations, at least to produce those vaccines like coronavirus where the international system cannot satisfy the demand. So I believe there's a special question in here for Dr. Blyden and Professor Pontiano. So if you really understood the question, you could go ahead and answer within 2 minutes.
I think that the issue of whether vaccines can be made in Africa or not hasn't really been addressed. We've just accepted that it's difficult, it's challenging, or it's not possible because of all of these, many of these factors that we've heard about just now, like cold chain and so forth. But the reality of the matter, what I've tried to do is not so much develop a vaccine, but develop a way in which vaccines and other products can be made in Africa. That's what the focus needs to be. In fact, as it turns out, not all vaccines need cold chains. It's possible to develop room temperature vaccines with modern technology. Not all vaccines need to be intramuscularly injected. If you're making a vaccine, you can make intranasal vaccines that you simply spray up the nostril, and it can stimulate an immune response. and a variety of other pathways, inhaled vaccines, sublingual vaccines under the tongue, et cetera, et cetera. What we've seen in this pandemic is that the emphasis has been on intramuscular vaccines, which are obviously very complex and complicated to make, especially if you're going to make something like an mRNA vaccine, which is essentially made with chemistry. It's not made with biology. And so I think that what we need to focus on in Africa is identifying enabling technologies like the ones I mentioned today, and there are others, that will allow us to make vaccines under the conditions that prevail in our particular locations. And if those are all part of a network, then we will be able to support each other with the expertise and the resources that we need in Africa under arrangements such as the free trade agreement and so forth. And we shouldn't find ourselves in a situation where we can't get the medicines we need to protect our populations.
Professor Pontiano, can you also come in with the question that was addressed to you concerning how education and environmental quality and how serious it has effect on our healthcare delivery? Dr. Anthony Donubari Enwin wants to find out how local conditions affect disease variants, especially considering hygiene and climatic variables.
Okay, let me take it that disease variants is talking about the SARS-CoV-2 variants that we've been talking about, and the local conditions like hygiene and climate affect the variants. I think to me the most important way to address the variants, if we don't want newer variants, is to reduce our transmission through different ways. Vaccinations, one of those. The herd immunity, Isabella was talking about. If we reduce on transmissions, we're unlikely to have additional variants. I think that's very key. On the other hand, there are those who have also argued that maybe if we have transmissions, we may naturally, it may lead us to a more mild SARS-CoV-2 that we may be able to manage. So I think apart from that, other climate or hygiene and the rest may not be as important as really how do we reduce on transmissions so that we don't have the virus spreading and mutating. That will be a good way of addressing that. There are many unknowns whether it's good to have some transmission, to have, as I've said, a more mild one. There's also a question about traditional herbs and the COVID-19. Of course, we encourage traditional medicine, but we are saying that even traditional medicine should be done in a scientific way. The only way Africa and many, even here in Uganda, we've been talking about it. Last week, we had a WHO team to talk about traditional medicine and COVID, is to have scientific approach to looking at traditional medicine. The principles of doing good research are important. That will lead us to really addressing, finding out what works. If we don't do that, then we delay, this works, doesn't work, you delay, sometimes you waste resources, people's expectations, but traditional medicine should be encouraged, but do it in a scientific way. If it's going into preclinical studies, do the preclinical studies. Capacity should be built for that. If you're going to clinical trials, animal studies will be nice, small phases and you do a very good, the problem are resources, a good randomized controlled study for some people to get the medicine, others don't get it, and see what happens afterwards. So indeed have a medicine as a role, but we have to use it in order to get the best out of it, use good science. Thank you.
Professor Pontiano Kalibu and Dr. Elumeno Richard Blyden for your support, and to you, Isabella Tayanaka, for your presentation as well. Imelda, Giselle Amuji for your support as well, and to all our cherished participants who made time to join us on this impactful, transformational episode of the COVID-19 Diary, looking at vaccine development, variant, and herd immunity. I think the point is made clear with what our speaker said, that we have to always stay attentive, stay very protective for ourselves, because COVID-19 isn't gone entirely, but there should be conscious efforts to keep ourselves safe. But let me take remarks from my colleagues. Belle, what is your view on your take on today's impactful presentation so far?
Okay, so as we've already learned, we don't know whether there's going to be more variants coming up, and we don't know whether the vaccines are going to be very effective to stop us from transmitting the virus to others. But then what we know is that the vaccine can help us by stopping us being, should in case we are affected with it again, we are not going to feel the symptoms, we are not going to fall sick. It's going to protect us from getting sick the second time we are exposed to the virus. So it's advisable that as we've been vaccinated, we keep on wearing our nose mask, we keep on giving the space there, the 5 meters distance, we keep on washing our hands with soap and water and the running water, and then we keep on sanitizing our hands.
Mrna, your views?
Okay, I actually wanted to ask a question about the herd immunity. I wanted to know why we are taking the boosters. And in Bella's presentation, she actually made mention that we don't know how long a vaccine, the COVID-19 vaccine, is going to stay in our body. So I'm tempted to take that as an answer. But I would like in the future series, I would like us to break down so that we will know how long. By then, I hope the scientists come out with their answers and the reason why we should take the boosters. Yeah, that was what I really
actually, if I'll answer your question, you see, there are more variants coming out, especially the Omicron. Yes, the first, let's say Pfizer, for instance, their vaccine, AstraZeneca, we know it prevents us from getting sick, but then it cannot prevent us from transmitting the virus to others. At the same time, some of the variants are stronger than the virus itself. Okay. So you take the booster so that it also prevents you from getting sick when you are infected with the Omicron variants. Do you understand? That is why some of the vaccines you take once because it can immune you for a very long time. Some you take twice because the first shot can immune you for a short time, and then you take the second time that will immune you for a very long time. But the boosters are to protect you from getting sick when you are infected with the new variants like Omicron or Delta, which the first vaccines cannot protect you from getting sick.
Okay. You know, it's been amazing. I have just 5 minutes to sign out, but I would want to show this very quick one to Dr. Blyden from Mary Mwangi. And she says, is there a potential for developing edible vaccines against COVID-19? Dr. Blyden, can we achieve this?
Well, I think any of the variety of vaccines that are known to man could be developed into a vaccine specific for COVID-19. The amount of investment and effort that has gone into that to exploring different types of vaccines has not been that great. So in effect, what we want to see from the African community, especially of academic researchers and so forth, is exactly those sorts of ideas of different ways to deliver and induce immunization in the person taking the vaccine. And then it comes to people like us who are trying to develop manufacturing to be able to manufacture it locally. And then it would go to people who are experts in doing clinical trials to do the clinical trials and establish the efficacy and the safety of the product. And then if it's approved by our government and the WHO, then it can be manufactured and become available. I'm actually designing an edible vaccine at this time. Well, it's actually not edible, it's sublingual, but it's almost the same thing, because if you put it in your mouth, I guess in principle you're eating it, right? But the answer in short is yes, I believe it's possible.
To much more innovative ways of fighting COVID-19 in the African continent. But as the time holds, we ought to be safe, get vaccinated, and then keep following the COVID-19 protocols, your nose mask, social distancing, and hand washing. Everything still applies because we need you alive. So until we come your way with the next edition of the COVID-19 Diary, we believe that you had a great time with us on the 10th edition of the COVID-19 Diary, and do well to hit on your social media platforms and share the wonderful information you have gotten from us, especially to the science journalists who have joined us here. Do well to refer to the COVID-19 Diary on AAU TV as a contributor to the information you have, and always keep us in your books as a great partner to share with you the insights. I am Mrna Chudako, and I was joined here by Imelda Giselle Mudi and Isabella Tahenakwa, co-panelists and science journalists representing from the Association of African Television. We hope you had a great time. Have a nice day, and bye.